All right, so our slides, the bacteria that we have diluted out into that water has dried on this slide. So now what I'm going to do is I'm just going to use the Bunsen burner. I'm going to do a really quick heat treatment for it. So let me light the Bunsen burner using your striker. Okay. And this is actually quite fast. It's a pretty quick process. So I'm going to take my tongs and I'm just going to run the slide through the flame for just a couple seconds. And what this is doing, it's called heat fixing, or we're fixing our bacteria onto the plate so that as we wash these different solutions through it, it's not going to come off. Okay, these cells aren't just going to wash right off. We have a few slides to do this with. And I've already done that with the other slides. So that's pretty much it. It's a really quick process. So now that we have that, I'm going to show you how to do gram staining. So let me just change the camera here. So we have our couple slides, and I'm just going to show you using two of them how to do gram staining. It's a fairly quick process. We have several different reagents here. We're going to start with crystal violet. You can see that this is crystal violet. That's going to be our first reagent. I always think of it C-I as. I don't know why, it's just kind of something that helps me remember the order. So C for crystal violet, I for iodine, this is grams iodine, A for alcohol, and we're going to be using 95% ethanol. And then S is for safranin. This is our counter stain. All right, and I want to make sure that I have different droppers for each reagent. I don't want to be mixing any of these accidentally because I'm using the same dropper for each. So I have one for iodine, and then I've got some disposable ones. I did label them. Sometimes it helps to just with Sharpie kind of put the letter of whichever reagent or even the whole name so that you know which dropper goes with which, so you're not accidentally kind of mixing them up. And then it also helps to have them kind of set up right next to your reagents. Again, it just kind of helps so that you don't get confused on which pipette, which dropper goes with which reagent. I also have a staining tray here. And so this has just kind of like a little placement area here where your slide just kind of sits on top. And then as you flood it with your different reagents, the rest of it just kind of falls into this tray and collects it. You also want to make sure that you're close by a sink because we're going to have to rinse off each reagent each time. All right, so I'm going to just put it in my staining tray right now. These stains, I mean, they're called stains for a reason. You always want to make sure you got gloves on, apron or lab coat, and your goggles on. And it actually, you could hold it in your hand and use the stain, but I'm going to use this handy little tray because we have it. All right, so I'm first going to do crystal violet. This actually has its own dropper with it, so I'm going to use that dropper. If it doesn't, again, you want to make sure you have your own pipette dropper. So where I'm going to be adding this is directly to that center where we added that circle. I'm just going to do a couple little drops there, and it's covering, I don't know if you can see this, but it's covering that entire center region where we have our grease pencil outline. So I know that the bacteria are within that grease pencil. Now I'm going to wait a minute. All right, so we're going to pause here. A minute goes by. Okay, that's been a minute. I've got my timer here. So after that 1 minute, now I'm going to rinse it off. Now you could just hold this in your hand if you want to make sure you don't get stuff all over your hands. You could use the tongs. I'm going to turn the water on low. We don't want to be, ferociously rinsing this off. So I'm just going to turn my sink on low. And I'm just going to be rinsing off, and I'll try to catch the residue in this tray because why not? I'm going to just rinse off my slide. I actually kind of prefer to have the water hit the slide above where our bacterial stain is because I don't want the pressure from the water falling to be what's causing this bacteria. If there is any bacteria that wash off, I don't want it because of the pressure of the water. So it's just kind of lightly, gently rinsing it off. Now you can see that, hopefully you can see that that's nice and colored. Like I've got a pretty dark stain there, dark purple. So right now, every cell in the slide is stained purple, but it's not going to stay that way. So remember C-I as, so we did the C, now we're going to do the I. This is the mordant, this is iodine. So we're going to add iodine, same like we did before, just a couple little drops right into the center. And then we're going to let that sit for a minute. And what this is doing, so I've got my timer, stopwatch. Set it for a minute. Now what this is doing is it's basically binding that crystal violet to our basically the cell wall on our positive cells. So the gram-negative cells, it's not really doing a whole heck of a lot to. It's basically holding it onto that outer cell, our thick cell wall on gram-negative, or I'm sorry, gram-positive cells. Right now, it's kind of binding that crystal violet stain to that thick cell wall. On our gram-negative cells, remember we have that lipid layer, so it's really not able to get through to bind it to our cell wall. All right, so again, we're going to wait a minute. Okay, it's been a minute, checked our timer. And now we're going to be rinsing this off using a gentle flow of water to rinse off that iodine. And I typically like to make sure that I'm not dropping water directly onto my slide where my bacteria is. I kind of want it to flow above it so that it just kind of gently rinses down the location where our slide is. Or I'm sorry, where our cells are. All right, so just kind of a quick water rinse. And now CI as, C is done, I is done. Now it's time for the A. This is the alcohol. So we're going to use ethanol. Now this is not for a full minute. This one's only for about 10 to 30 seconds. It kind of depends on which procedures you're following. I typically like to do this for just about 10 seconds. So what this is going to be doing is it is essentially like, I don't want to say the word dissolving, but it's essentially breaking apart that lipid membrane on our gram-positive cells, our gram-negative cells. So that outer lipid membrane on our gram-negative cells, this is essentially kind of creating a little bit more of a porous type of cell lipid membrane. And the purpose of this, that's been about 10 seconds, I'm going to rinse it off with water. Purpose of this is so that when we do our counter stain, our safranin stain, it will be able to actually stain these cells. So remember, that lipid layer is kind of preventing that stain from getting through. Well, now we just kind of disrupted that lipid outer layer. So now whatever stain we add, it will stain. So I'm going to add our safranin. So again, just a couple quick 2 little drops right on the center of where our cells are located within that grease circle. And set the timer. 1 more minute. Okay, minute's up. We're going to rinse off our safranin. And gentle water flow. And when we are rinsing off our reagent, I don't like to have the water fall right onto the center of where our cells are. I kind of like that letting just a little bit above it and then just gently rinsing down the slide where our cells are. And I don't want the water pressure to sort of forcefully remove some of our cells. Now they are heat fixed on there, but that doesn't mean they're going to be there forever and ever and ever. In a perfect world, they would be, but if you're adding quite a lot of pressure, that might actually cause an issue. All right, so now I've got a pretty wet slide, but it's fully stained. So we are just going to blot it now. I'm just going to take some paper towels, kind of a few layers of paper towel, place our slide on there, and then just gently press down on it. Not too hard. We don't want to go back and forth. We don't want to smudge this because, again, there are some cells on there, hopefully at this point, that are stained, and we don't want those to dislodge. And they are heat fixed, but that doesn't mean they're going to be like indestructible. So we've got our stain. That's sort of what it looks like. And it should look kind of, you could see a little bit of blue, purplish on it still, but those are our stained cells. So now what we're going to do is we're going to go over to our compound microscope, and I've got a digital microscope setting set up so you'll be able to see what's happening within the field of view, and we'll hopefully get a look at whether this is Bacillus megaterium. We're going to see what kind of shape, what kind of arrangement they are, and what type of gram staining it does, positive or negative. All right, I'll get that set up in a minute.